Good morning, everybody. Welcome to day 2 of the MMBioS 2022 Cell Modeling Virtual Workshop. And today, I'm very happy and excited to be teaching you about MCell and Cell Blender, the tools developed in MMBioS that let us model not just biochemical reactions, but biochemical reactions with diffusion, so-called reaction diffusion. And you're going to learn today about the tools we've developed that make this possible based on MCell Monte Carlo cell that tracks the individual particles of molecules as they diffuse and interact with each other in realistic 3D geometries. So the motivation behind the original development of MCell was within my research group and my colleagues and my mentors over the years. How do synapses work and how does synaptic structure influence synaptic function? And especially complex in this are not just the reaction pathways, which are of course extraordinarily complex, but the 3D space in which synapses exist in the brain, a space called neuropil. And I put 3 exclamation points after this because the word neuropil strikes fear. You know, it's such a crazy space, the extracellular space. So we need to be able to model that. We need to model the organization of the cytoplasmic organelles that exist in synapses. We need to study and include the distributions of receptors, ion channels, enzymes, transporters, et cetera, on those membranes. And then, of course, the biochemical reaction networks that govern those interactions. And after all this, it's easy to see that cells are not well mixed. And because synapses are so small, cellular compartments between and among organelles are so small, those volumes are so small, the numbers of molecules can be quite small. So that means the simulations must include the stochastic spatial temporal dynamics. So not just space and time, but also small numbers of molecules that cause these fluctuations. In the laboratory of Mika Salpeter at Cornell University in the mid to late 1980s, we developed Monte Carlo simulation technique that later, after I joined Terry's lab, we named MCell for Monte Carlo Simulator of Cellular Microphysiology. We invented this technique that is able to model the realistic 3D reaction diffusion systems. We use Monte Carlo methods to track the Brownian dynamics diffusion and reactions of individual particles as they diffuse through 3-dimensional volumes, and also molecules embedded into surfaces and their diffusion on those surfaces. And because this is all done with particles and not with differential equations, we can easily handle arbitrarily complex 3D geometry because the geometries don't need to be tetrahedral meshes that are very well constrained and behaved, which is really, really hard to, those structures are classically really, really hard to generate and takes a great deal of expertise. And there's a whole field of study for creating those geometries. In MCell, we can get away with just having surface meshes that are triangulated, which is a much easier problem to solve. And there's good software available for doing that. And because we are dealing with individual particles, we can also handle arbitrarily complex reaction networks, either as Markov processes, explicitly written Markov processes, which of course would be what we learned yesterday as a as a reaction network, but we can also handle network-free rule-based specification using BioNetGen language. And these are modeled as discrete event driven point processes using the network-free method that Jim already introduced yesterday. And all of this is possible and due to a flexible and powerful Python API. For people who are computer modeling experts, they can make great use of this powerful Python API. And over time, you can train yourself and become such an expert. And with your drive to, for your personal research reasons, with your drive to make computer models, that is a really good motivating factor to learn more and more Python, which I encourage you to do. However, we also have a sophisticated model building, visualization, and analysis environment called Cell Blender, which if you don't want to deal with a lot of writing pure Python code, Cell Blender hides that from you and lets you still create really powerful models using this visual programming environment that I'm going to teach you today, and create really sophisticated models all visually without touching any Python. And then later, as your needs expand, you can use Cell Blender and the Blender add-ons that we have built into Cell Blender. Start adding Python code to customize your model to make it do fancier things, but in a more structured and gentle way than just starting with Python from scratch.